Hi everyone, welcome to my presentation today on antibodies. So today we're going to be exploring one of the most essential and fascinating components of our immune system, antibodies, also known as immunoglobulins. So in this video, I'll walk you through the structural features of an antibody and how these features correlate to its function in the immune response. So to start, let's talk about the immunoglobulin superfamily. So this is a large group of proteins that play critical roles in immune defense, cell recognition, and signaling. Now, members of this superfamily include antibodies, T cell receptors, MHC molecules, and more. So what connects them is their shared structure, immunoglobulin domains, which are globular regions that are stabilized by disulfide bonds. So these domains provide the framework for diverse but related functions in immunity. So for this presentation, I've created a model of an antibody which outlines some of the key structural features of it. Now, as I walk you through the model, I'll describe each part, as well as its significance and how it contributes to the overall function of the antibody. Now, even though my model is fairly simple, it represents the core components that are necessary to understand antibody structure and function. Now, specifically, I'll explain the heavy chain, the light chain, the hinge region, disulfide bonds, the paratope, the complementarity determining region, and how the antibody is divided into FAB and FC fragments. We'll also discuss how secreted antibodies differ from B-cell antigen receptors, and also why this distinction is important. And finally, I'll end with a quick summary of the functions of antibodies. Now before we dive in, I want to say again that my model is fairly basic, right? It's built on a flat surface, so I'll be pointing to the different parts as opposed to picking it up because I don't want to deform the Play-Doh that it's made from. I've labeled some of the key features like the heavy chain and the light chain and the hinge region, just to kind of make it easier to follow. Some of the regions aren't explicitly shown in the model, but I try my best to explain the area which these regions are found. So not to worry, I'll make sure things are crystal clear as we go along. All right, now let's head on over into it. All right, so let's dive into the model I created to represent the structure of an antibody. So as you can see, this is a simplified version of an antibody with its core components highlighted. So antibodies are Y-shaped molecules, and their structure is critical to their function in the immune response. So let's start with the heavy chain. So each antibody has two identical heavy chains, which I've represented here with the orange Play-Doh. All right, so this one over here and this one over here. Now, the heavy chain is the backbone of the antibody, and each chain contains about roughly 440 amino acids. Now, structurally, the heavy chain has one variable domain at the top, which is this area here, and three constant domains below it. So they're typically labeled CH1, which would be here, CH2, which would be here, and CH3, which would be here. So next we have the light chains, which I've represented here with the purple Play-Doh. So there are two identical light chains, so each with around roughly 220 amino acids. So each light chain has one variable domain right here and one constant domain, which is right here, the lower part. Now together with the heavy chain, the light chain contributes to forming the antigen binding site. Now let's focus on the variable regions. So these are located at the tips of the Y shape where the heavy and light chains meet. So right up here. Now I've added an indentation here to represent the paratope, which hopefully you can see it from this angle here, right? These little grooves right there. I've included those indentations, like I said, to represent the paratope. And this groove is the specific site that binds to an antigen. So I've kind of created a little antigen model here with the pink Play-Doh just to show how it fits directly into that groove there. And that's what constitutes that paratope, okay? So let's remove our antigen out of there and get back on subject here. So this is the region that is unique for each antibody, allowing it to recognize a specific antigen. So next, let's talk about the hinge region. So I've labeled it right here in the middle where the arms of the Y connect to the tail, right? So this is the flexible part of the antibody, which allows the arms to move and adapt so the antibody can bind antigens at different distances. Now, these bonds stabilize the antibody by linking the heavy chains together and connecting the light and heavy chains. Now, without these bonds, the antibody would lose its shape and functionality. Now, within the variable regions of the heavy and light chains, we find the complementarity determining regions, or the CDRs. Now, these are hypervariable regions located within the paratope that directly interact with the antigen. So, although it's not explicitly shown on this model, they're responsible for the antibody's specificity. Okay, so that's important to note. Now, moving right along, let's break the antibody down into its functional fragments. So, the Fab fragments, or fragment antigen binding, it consists of the variable and constant domains of both the heavy and light chains. Right, so that would be right here. Now, these are the arms of the Y, and they're responsible for binding to antigens. On the other hand, the FC fragment, or fragment crystallizable, is the tail of the antibody. So the FC fragment interacts with immune cells and the complement system to trigger an immune response. Now, let's address the difference between secreted antibodies and B cell receptors, or BCRs. Now, secreted antibodies like this one here are free floating in the blood and lymph. Now, they actively neutralize pathogens and mark them for destruction. So BCRs, however, are membrane-bound versions of the antibodies on the surface of B cells. So they have an additional transmembrane domain, which would be somewhere down here, that anchors them to the cell, allowing them to transmit signals when they bind to an antigen. So it'd be an additional domain down here that just kind of affixes this to the surface of the cell. Now, this distinction is essential because BCRs activate B cells to produce more antibodies and start the immune response. So finally, let's summarize the key functions of antibodies. So they neutralize pathogens by blocking their ability to infect cells. So they opsonize pathogens also to mark them for destruction by phagocytes. Additionally, they activate the complement system and facilitate antibody-dependent cellular cytotoxicity by recruiting natural killer cells. So all of these functions depend on the unique structure of the antibody, as we've just discussed. So all things here being discussed, it's easy to see that antibodies are truly fascinating molecules that are vital to the immune system's ability to protect us from disease. So by understanding their structure, we gain a deeper appreciation for how they work to keep us healthy. So I hope this presentation has helped to clarify the essential features and functions of antibodies. And I thank you for watching.